Good day, everyone. Today, we are going to begin with an important subject pharmacognosy and phytopharmaceuticals. The topic for today is the introduction, which will give you a strong foundation for understanding natural drugs and their role in pharmacy. Let us start with the basic definition. Pharmacognosy is the branch of pharmaceutical science that deals with the study of drugs obtained from the natural sources such as plants, animals, minerals, and microorganisms. The word itself comes from the Greek terms pharmakon, meaning drug, and gnosis, meaning knowledge. So, pharmacognosy essentially means the knowledge of drugs of natural origin. Historically, pharmacognosy is one of the oldest branches of medicine. Before the development of synthetic drugs, humans depended entirely on natural substances for the treatment of diseases. Even today, a large number of modern medicines are either directly obtained from natural sources or are derived from compounds originally found in nature. This shows that pharmacognosy continues to play a very important role in modern healthcare. Now, let us understand the scope of pharmacognosy. This subject is not limited to just studying plants or crude drugs. It includes identification and classification of natural drugs, cultivation and collection of medicinal plants, extraction and isolation of active constituents, and evaluation of drug quality. It also involves the study of traditional systems of medicine and the development of modern herbal formulations. So, pharmacognosy acts as a bridge between traditional knowledge and modern pharmaceutical science.
Next, let us discuss the sources of drugs in pharmacognosy. Natural drugs are obtained mainly from four sources. The most important source is plants. Different parts of plants are used as drugs, such as leaves, roots, bark, seeds, and flowers. For example, dish chalice is obtained from leaves and is used in heart conditions, while cinchona bark is a source of quinine used in malaria. Plants produce chemical substances known as secondary metabolites, which are responsible for their therapeutic effects. The second source is animals. Some drugs are obtained from animal sources, such as hormones, enzymes, and oils. For example, insulin was traditionally obtained from animal pancreas. The third source is minerals, which includes substances like iron and calcium that are used in medicinal preparations. The fourth source is microorganisms, which are very important in the production of antibiotics and enzymes. Now, let us understand what crude drugs are. Crude drugs are natural substances used in their raw or unprocessed form. They may undergo simple processes like drying but are not chemically modified. Examples include dried leaves, roots, and bark. These crude drugs contain active constituents, which are the chemical compounds responsible for the therapeutic effect. These active constituents are very important in pharmacognosy. They include different classes such as alkaloids, glycosides, tannins, volatile oils, and flavonoids. Alkaloids are nitrogen containing compounds with strong pharmacological activity. Glycosides consist of a sugar part and a non sugar part and are important in cardiac drugs. Tannins have astringent properties. Volatile oils are responsible for aroma and therapeutic effects, and flavonoids are known for their antioxidant activity. These compounds are isolated and studied to understand their effects and to develop medicines. Now, let us move to phytopharmaceuticals. Phytopharmaceuticals are modern plant-based medicine that are standardized and contain defined amounts of active ingredients. Unlike crude drugs, which are used in raw form, phytopharmaceuticals are scientifically processed and evaluated for quality, safety, and efficacy. This makes them more reliable and consistent in their action.
Another important aspect of pharmacognosy is the evaluation of crude drugs. This is done to ensure their quality and purity. Organoleptic evaluation involves using our senses to identify characteristics like color, odor, and taste. Microscopic evaluation involves studying the cellular structure of the drug. Chemical evaluation helps in identifying active constituents, and physical evaluation includes parameters like moisture content and ash value. All these methods helps in ensuring that the drug is genuine and of good quality. We should also be aware of adulteration of crude drugs. Adulteration means the substitution or mixing of inferior or harmful substances with genuine drugs. This may happen intentionally for profit or accidentally due to the lack of knowledge. Adulteration reduces the effectiveness of the drug and may even cause harmful effects. Therefore, proper identification and evaluation are very important. The quality of crude drugs can also be affected by several factors such as climate, soil conditions, time of collection, storage, and processing methods. These factors influence the concentration of active constituents and ultimately the effectiveness of the drug. Pharmacognosy is closely related to several other sciences. It is connected to botany for plant identification, chemistry for studying chemical constituents, pharmacology for understanding drug action, and microbiology for studying microbial products. This makes it a multidisciplinary subject. In recent years, there have been many modern developments in pharmacognosy. These include standardization of herbal drugs, use of biotechnology such as plant tissue culture, advanced extraction techniques, and the development of phytopharmaceuticals. All these advancements are helping to make natural drugs more effective, safe, and acceptable in modern medicine. So, to conclude, pharmacognosy is a study of drugs obtained from natural sources and plays a crucial role in both traditional and modern medicine. It involves understanding the sources, active constituents, evaluation, and application of natural drugs. With the growing interest in herbal medicines and natural products, pharmacognosy continues to be an important and evolving field. Thank you.